In this video, we'll spend a few minutes looking at the development of the brain, the embryonic and fetal development of the brain. Now, we're going to start with this overview and just run through some basics, and then we're going to have a look at something called neural tube formation, which is really the start of the formation of much of your nervous system. So as we look here, we can see that there's a structure that we're going to talk about, which is called the neural tube. And it's from the neural tube, and not surprising with a name like that, it is a tube, and it does contribute to the nervous system. This tube is going to form three, basically, vesicles, some swellings within it. They're going to become the forebrain, the midbrain, and the hindbrain. And we can see here, so actually what we do is we start off with a long tube structure, and the tube structure then undergoes, basically it ends up with swellings within it, and then it's going to change shape. It's going to fold on itself and bend in a few places. So we're going to start off with that neural tube. Now let's start talking about that neural tube, and I'm going to switch to this drawing. And let me try and explain what we're trying to see here in a two-dimensional realm, which is actually a three-dimensional structure. So when we have embryonic development, we start off as one cell and then many cells, and then cells differentiate and cells start to form masses of cells and structures. And there's one point in embryonic development, and we're not going to get really bogged down in the different names of the different stages, embryonic development and the processes. But what we have is basically a sheet of cells. So we have a top sheet of cells here. We have a bottom sheet of cells, and then a group of cells that are in between, that are stuck in between. And what I'm trying to do over here is show it in sort of a cross section, that you can see the top, the bottom, and the cells that are in between. And these are the three primary germ layers, the ectoderm, the mesoderm, and the endoderm. Well, how do I get from something like this? Because, in many ways when I look at this, I always think this looks like a bit of like a pita sandwich, when you have that pita bread open, you put stuff in the center. That's what it's kind of like. That one layer is one layer of the bread, the other is the opposite, and the filling is the middle. Now, how do I get from a flat structure, because it's this ectoderm here that's going to form the neural tube. So it's this area, and you see it's an elongated structure, and I'm trying to show it heading off into the distance here, but that's the cut edge. Well, let's just sort of start looking at that cut edge. Now, when that sheet first starts out, the sheet, and I'm just right now just looking at this top sheet right there. Okay, this. I'm looking at sort of edge on. This top sheet, what happens is we're going to start to have these things called neural folds. So part of the sheet is going to kind of bend up, bend down, bend up, etc. So I'm going to end up with these folds in it. So let's now draw that. So we're going to draw it with the folds in it. So it goes from that to start to get this shape. Okay. Now these folds are going to keep growing, and the folds are going to actually grow towards each other. And eventually what happens, not surprisingly, is that as the folds keep growing, they're actually going to touch. So, as I said, we start off with a flat sheet. It started to develop some folds, and those folds eventually, the two edges of the folds on each side came together and formed this. Well, all you have to do now to actually make a tube is you have to come along, and you have to basically, all you have to do is separate that material, and all of a sudden now I have a tube. And so we end up with when these folds form, okay, it's going to happen all the way down. So we're going to have this groove that's going to form. And eventually the tube will form out of it and be under there. And that's the neural tube that we're talking about. So eventually, as I said, this will fold up. Eventually I have a tube that's going to form and is going to run underneath here. And it's derived from this top layer. All right. So once I get the neural tube, that's the first step. Now, parts of the neural tube will go on to specialize to become different parts of the brain. And we can see the flow chart here is that the neural tube first gives rise to the forebrain, the midbrain, and the hindbrain. And we can see the tube has already started to bend, and we're starting to see these swellings. So the caudal end is becoming the forebrain, and you can see it's also color-coded. The blue is the midbrain, and this is going to be the hindbrain. And during development, it just keeps elaborating a bit more. Now it's going to take on different names because the forebrain is going to divide into two things called the telencephalon and the diencephalon. Sometimes they say encephalon. It depends whose pronunciation you want to follow. We also have the midbrain, which then becomes the mesencephalon, and the hindbrain becomes two different regions, which are the metencephalon and the myelencephalon. And you can see how we've gone from this region is now split into two different regions, etc. So you can really follow the words, the colors, and start to really get a sense as to what's happening. Well, not surprisingly, more embryonic development happens, fetal development happens, and we really start to elaborate the brain. So this first bit that was forebrain becomes the telencephalon, and if you come here, you can see the color comparison. That really, the telencephalon really makes up most of what people know as the majority of the brain, which is the cerebrum. The diencephalon, okay, stays buried within here as part of the diencephalon or diencephalon, some people would say. And then also when we look at the metencephalon, the myelencephalon, those structures are really becoming part of the brain stem. And that, of course, is broken down into the midbrain, the pons, and the medulla. And you can start to see here's the medulla down here, and above it is the pons. You can tell the pons because the pons sort of bulges out here. And then above or superior to the pons, we get into the midbrain region. So that's sort of a very quick overview of how we go from a neural tube and we fold the tube, we get the tube to swell out. Now, I want to point out within this tube, so let's draw the basic tube. Once we form this tube, right? So this tube has some thickness to it, some material to it, okay? It's this material. It's the materials of the walls of the tube that are going to become these structures. So all the way along here is elaborates. It's that. Now, if you reflect back on previous courses, and I did mention the concept of ventricles in a previous video, where do the ventricles come from? Which are those open spaces where CSF flows through? Well, this empty space within the inside of the tube is really going to be elaborated and become part of the ventricles. Also, when we quickly talked about the spinal cord has a canal that runs right down the center of it, which is called the central canal. And of course, this space is seen in the central canal of the spinal cord. And once again, this space is eventually the future home of cerebrospinal fluid and becomes a path that cerebrospinal fluid uses to travel around inside the brain and eventually on the outside of the brain where it's eventually reabsorbed. So that's a quick overview, just looking at some of the features. Now, I would make sure you go back, read the book, watch some of the videos on development, and really get a sense of it, because these names are very important. I know there's a lot of them, but knowing about telencephalon, diencephalon, metencephalon, myencephalon, because often they can be used when talking about parts of the brain and where it was derived from. Okay, we're going to end the video there.